evening, everyone. Today, we are going to focus on essential aspect of your A-level studies, mastering general insurance through multiple choice questions. Without further ado, let's get into it. Which statement about resolution and magnifications are correct? The answer is A. Why? Resolution refers to the microscope ability to distinguish between 2 points that are close together, while magnifications refer to how many times larger the image appears compared to the actual size of the object. The eyepiece reticule has a scale with 100 divisions. A stage micrometer has a scale with 50 divisions, and each of which is 0.04 millimeter apart. Using a multiply 40 objective lens, the whole length of the stage micrometer scale line up with 15 divisions of the eyepiece reticle. What is the actual length of the 100 division scale of the eyepiece reticle? So to find the actual length of the 100 division scale of the eyepiece reticle, you have to follow these steps. Step 1. Determine the total length of the stage micrometer scale, which is the stage micrometer has 50 divisions and each 0.04 millimeter apart. Therefore, the total length of the stage micrometer scale will be 50 multiply 0.04 millimeter, you get the value of 2. Step 2. You have to determine the length of the stage micrometer as seen through the eyepiece reticle. The stage micrometer scale line up with 15 divisions of the eyepiece reticle. Thus, the length of the stage micrometer as seen through the eyepiece reticle is equivalent to 15 divisions of the eyepiece reticle. Step 3. Calculate the length of 1 division of the eyepiece reticle. Since 15 divisions of the eyepiece reticle measure 2 millimeter, and the length of the 1 division of the eyepiece reticle would be 2 millimeter divided by 15, you get the value of 0.1333 millimeter. Step 4. Calculate the total length of the 100 division scale of the eyepiece reticle. If 1 division of the eyepiece reticle is 0.1333 millimeter, then the length of 100 division scale of the eyepiece reticle is 100 multiplied by 0.1333, you get the value of 13.33 millimeter. Rounding to a suitable precision, the length is equivalent to 13 millimeter. Therefore, the answer for these questions, which is the actual length of the 100 division scale of the eyepiece reticle, is closely to the opinion of B, which is 13 millimeter. A prokaryotic cell, which is 1 micrometer in diameter, is magnified 50,000 times in an electron micrograph. What is the diameter of the cells in the electron micrograph? To determine the diameter of the prokaryotic cells in the cells micrograph, electron micrograph, after magnifications, you have to follow this step. First, identify the actual diameter of the cells, which is the diameter of the cells is equivalent to 1 micrometer. And 2, step 2, you have to convert the actual diameter to millimeter, which is 1 micrometer is equivalent to 0.001 millimeter. And step 3, calculate the diameter in the electron micrograph. The cell is magnified 50,000 times. So diameter in a micrograph is equal to actual diameter multiplied by magnifications. And diameter in the micrograph is equivalent to 0.001 millimeter multiplied by 15,000 times. And you get the value, diameter in a micrograph is equal to 50 millimeter. So the correct answer will be C, which is 5 multiplied by 10 to the power of 1 millimeter. Number 4, a scientist carried out an experiment to separate cell structures in animal cells. The cells were broken open to release the cell structures. This extract was filtered into a centrifuge tube and then spun in a centrifuge. The heavier cells structures sank to the bottom, forming pellet 1 as shown in the diagram. The liquid above pellet 1 was poured into a clean centrifuge tube and spun in the centrifuge at the high speed to separate the next heavier cell structure. The cell structure sank to the bottom, forming pellet 2. This procedure was repeated twice more to obtain pellet 3 and pellet 4, each containing a single type of cell structure, which row show the order in which the cell structures were collected. The answer will be pellet 1 is nucleus, pellet 2 is mitochondria, pellet 3 is lysosome, and pellet 4 is ribosome. Radioactive level amino acids are introduced into a cell that is actively secreting an enzyme in which cell structures where the radioactivity first become concentrated. The answer will be A. What is the general formula for amylase? This is also a common question which already exists in October 2018. And the question is asking about general formula for cellulose. As you know, amylose and cellulose are linear polymer of glucose which is linked with 1,4 bond. The answer will be C. The diagram shows relationship between some important molecule and bond. What is represented by circle number 1, 2, and 3? Answer will be A. In unsaturated lipid molecule, where are double bond located? In unsaturated lipid molecule, double bond are located within fatty acid only, and the double bond are found within the fatty acid chains, which are part of the lipid molecules, and this double bond cause kink in the fatty acid chains, which affect the lipid structures and properties. Phospholipid and triglycerides are important biological molecules, and which properties are correct for this molecule? The answer, the correct properties for phospholipid and triglycerides are C. These can be explained through triglyceride are non-polar molecule because they consist of 3 fatty acids and glycerol backbone, which are all hydrophobic. And phospholipids have a partially hydrophobic nature because they contain both a hydrophobic tail, which is the fatty acid chain, and the hydrophilic head, the phosphate group, and attached molecule. The diagram shows 2 arrangements of amino acids in a protein, which row correctly names the bond at S and T. The answer will be A. Students were asked to highlight only the R group of 2 ring-shaped amino acids. Which pair of diagram are correct for both amino acids? The answer will be D. Which words from the table correctly complete the paragraph about enzyme? So the answer will be, when the pH of an environment is decreased below the optimum pH of an enzyme, hydrogen and ionic bonds between adjacent R group holding the tertiary structures are disrupted. And this is because changes in pH can affect hydrogen and ionic bond, which are crucial for maintaining the enzyme tertiary structure. The effect of substrate concentration of an enzyme catalyzed reactions are measured in 3 different conditions. Which statement is correct? Y is a non-competitive inhibitor which binds to a site other than the active sites of the enzyme. Which statement about the cell structure membranes are correct? So the correct statements about the cell structure membranes will be, channel proteins allow water-soluble ions and molecules across the membrane. Channel protein provide a pathway for ions and other water-soluble molecules to cross the membrane, and this is true. Second, glucose can pass into the cell via carrier protein. This is also true because glucose is transported into cell through specific carrier protein called glucose transporter. Number 3, oxygen passes freely through the membrane as it is soluble in lipids. This is also true statement because oxygen is a small non-polar molecule and can be diffused through the lipid bilayer of the membrane. Some glycoprotein act as antigen. It's also true because glycoprotein on the cell surface can serve as antigen, which are recognized by the immune system. Which of these features increase the efficiency of iron uptake by a root hair cells? The features that increase the efficiency of iron uptake by a root cell are 1 and 3. Why? Because can be explained through many mitochondria in the cells. This is true because mitochondria provide the ATP needed for active transport processes, which are essential for efficient iron uptake. And number 3 is also true, protein carrier in the cell surface membrane, because this carrier facilitate the transport of iron into the cells, which is crucial for efficient uptake. And a high concentration of iron in the glucose statement 2 is more related to storage rather than uptake efficiency. Number 16, what can increase the fluidity of the cell surface membrane at low temperature? The fluidity of the cell surface membrane at low temperature can be increased by the following factors. 1, double bond between carbon atom in the fatty acid change. Double bond create kink in the fatty acid change, which is prevent them from packing tightly together, which increases membrane fluidity. Cholesterol. This is also true. At low temperature, cholesterol help to prevent the membrane from becoming too rigid by maintaining space between phospholipid, thereby increasing fluidity. And statement 3 is also correct, that which is one of the factors that contribute to the fluidity of the cell surface membrane at the low temperature can be increased because shorter fatty acid chain reduce van der Waals interactions between the chains, which also contribute to increased fluidity. So the correct answer will be 1, 2, and 3. 17. The diagram shows the shape and the size of the two types of the cells. Which statement is correct about the palisade cells and epithelial cells shown in the diagram? The given answer will be the surface area to volume ratio is greater in the volume epithelial cells than the palisade mesophyll cells. A student observed the cells in the growing region of an onion root and obtained the data shown, which a percentage of cells contain chromosomes that appear as two chromatic. To find the percentage of the cells in each stage of the cell cycle, you can use the following formula. Number of cells in the stage divided by total number of cells multiplied by 100. Let's look into the interface percentage, where you have the number of cells will be 886. 886 divided by 1000, you get the, and multiply by 100, you get the value of 88.6%. Phosphase, you have 73 number of cells, where you have to divide it by 1000 multiplied by 100, you get the value of 7.3%. Metaphase, number of cells is 16, you have to divide it by 1000 multiplied by 100, you get the value of 1.6%. Anaphase, number of cells is 40, which is 40 divided by 1000 multiplied by 100, you get the value of 1.4%. And telophase percentage is 11, where 11 divided by 1000 multiplied by 100, you get the value of 1.1%. And based on this calculation, the percentage of cells in the prophase stage is 7.3%, and which matches the option A. Which of these events are part of mitosis? This is also a common question which is already exists in May 2019 and November 2023 examination paper. Telophase is one of the phase in mitosis, and it is a final stage. before cytokinesis, where the chromosome reach opposite poles of the cell and begin to decondense. An interphase is not part of mitosis itself, but rather the phase where the cells grow, replicate its DNA, and prepares for division. The answer will be telophase only. During which phase of the cell cycle does DNA replicate take place? The DNA replication occurs during the S phase of the cell cycle. So the answer will be G. 21. Which row is correct for guanine? Guanine has a double ring structure. It is a purine and join its complementary base with cytosine and with 3 hydrogen bond. So the correct answer will be A. Mellison and Starr investigated DNA in bacteria. They grew bacteria in a medium with only heavy nitrogen until all the bacteria DNA was heavy. These bacteria were moved from a heavy nitrogen medium and cultured in a medium with only light nitrogen. Sample of bacteria was collected from the first generation in a medium containing light nitrogen, and their DNA was analyzed. Hybrid DNA contained both heavy and light DNA, which row show the percentage of heavy DNA strand and the percentage of hybrid DNA molecule in the first generation produced in a module containing light nitrogen. The answer will be C. Which is the correct DNA triplet on the original DNA template that codes for the amino acid histidine? The answer will be C. So to determine the correct DNA triplet on the original DNA templates that code for his, we need to understand how the genetic code is translated. This is included by the mRNA, codon, CAU, and CAC. To find the corresponding DNA triplet on the original DNA template strand, we need to convert this mRNA codon to the DNA template strand. The mRNA codon CAU pair with the DNA triplet GTA, where A pairs with the T and U pair with the A. The mRNA codon CAC pair with the DNA triplet GTG. Since his is included by CAU and CAC, the DNA template strand triplet that corresponds to the codon CAU is GTA. So the correct DNA template triplet on the original DNA template that codes for his is C. The photomicrograph show tissue in a stain transverse sections of the plant stem. Students are asked to draw 4 adjacent xylem vessels elements. Each drawing or drawn to the same scale is correct. The answer will be D. What is the functions of the hair, trichomes, and xerophytic leaves? The primary functions of hair on xerophytic leaves is trapping a layer of moist air. Trichomes help reduce water loss by trapping a layer of moist air close to the leaf surface, which reduces the evaporation rate and help the plants conserve water in arid environments. Which features of companion cells are essential to their function? To answer these questions, look at the number 1. They are connected by plasmodesmata to the sieve tube element. And number 3 is they contain a nucleus and mitochondria. Companion cells are connected to sieve tube element through plasmodesmata, which facilitate the transfer of nutrient and signal. And they also contain a nucleus and mitochondria, which are important for their role in supporting the sieve tube element, which is including providing the necessary energy and metabolic function. So the correct will be 1 and 3. The photomicrograph show a human blood smear, which will identify the label cells. 1 is actually monocyte, 2 is neutrophil, and 3 is lymphocyte. The diagram shows the changes in ECG, electrical activity in the heart muscle during 1 cardiac cycle, and this is called electrocardiograph. which electrocardiograph show a rate of 75 beats per minute. B. Which effect could be due to reduced concentration of carbonic hydroxide? A reduced concentration of carbonic hydroxide would most directly lead to hemoglobin concentration will decrease. This is because carbonic hydroxide help convert CO2 into a form that can be transported in the blood. With less carbonic hydroxide, it's not efficiently converted, leading to a decrease in hemoglobin. Which statement correctly compares blood plasma and tissue fluid in a healthy person? In a healthy person, the correct statement comparing blood plasma and tissue fluid is blood plasma contains more protein than tissue fluid. Blood plasma has a higher concentration of proteins such as albumin compared to tissue fluid. Tissue fluid is derived from the blood plasma but loses many of this protein as it moves out of the blood vessels into the tissues. The diagram shows three features found in tissue of the gas exchange system: ciliate epithelium, goblet cells, and cartilage. And the combination of these three will be P. Which tube of the gas exchange system could be presented at the position P in the diagram? The answer will be C. The diagram shows a magnified section of part of the lungs containing specialized tissue. Which row is correct for structures labeled 1, 2, 6? The answer will be C. A short-term effect of smoking is decreased blood supply to the finger and toe. Which component of cigarette smoke causes this effect? The component of cigarette smoke responsible for decreased blood supply to the fingers and toe is nicotine. Nicotine causes vasoconstriction, which narrows blood vessels and reduces blood flow to extremities like fingers and toes. The symptoms of two diseases are listed. Which row identify disease 1 and 2? Disease 1, coughing up blood, pain when breathing, loss of weight. And disease 2 is a shortness of breath, difficulty breathing out, fatigue. The correct identifications is D. Lung cancer often presents with symptoms such as coughing with blood, pain when breathing, and weight loss. Emphysema, a type of chronic obstructive pulmonary disease, COPD, is characterized by shortness of breath, difficulty exhaling, and fatigue. Which statement about a non-infectious diseases may be correct? 1 and 3 is correct. 1 correct because many non-infectious diseases such as genetic disorder result from mutation in DNA. 2 is incorrect because non-infectious diseases are not transmitted by insect vector, and this is a feature of infectious disease. And 3 is correct because some non-infectious diseases like certain genetic disorder can be inherited from the mother or father and passed to the child. So the correct answer will be 1 and 3. The diagram shows one way of testing the effect of antibiotic on bacteria. So we have a colonies of bacteria in the petri dish with nutrient agar containing bacteria. And in the middle part will be the dish of the paper soaked in antibiotic. And within 5 days, you can see the bacteria is growing. Diameter of the zone measured every day for 5 days. So you can see there is a lot of bacteria. The table shows the resulting of testing 5 different types of bacteria. Zone of less than 30 millimeter show the presence of resistant bacteria. And which statement can be supported by the data? So the answer will be only type 2, 3, and 4 of the bacteria show resistance, to the antibiotic, but not these other type of bacteria. You can see how the bacteria is growing. The answer will be only type 2, 3, and 4. 2, 3, and 4 for the bacteria show resistance to the antibiotic, not for type of bacteria 1 and 5. Some animals have gene that code for small peptide called catalydicin. This peptide kills a wide range of bacteria by attaching to lipid in bacterial membrane. So weakening or disrupting them, scientists have produced a synthetic version of the catalycidin that kill bacteria that are resistant to a number of antibiotics, such as tetracycline. Which pair of statements explain how the synthetic catalycidin might help with the problems of antibiotic resistance? The answer will be, it could be used instead of tetracycline, allowing tetracycline resistance to be reduced. And number 4, it could be used to kill multi-drug resistant strain of bacteria for which there is no effective antibiotics. Hybridomas are used as a basis for a production of large number of monoclonal antibodies. And which statement describes how hybridomas are made? Hybridomas are made by fusing B lymphocyte with cancer cells. Specifically, hybridomas are created by fusing antibody-producing B lymphocyte, not beta lymphocyte, with myeloma cancer cell. This function results in cells that can be produce a specific antibody from the B lymphocytes are divided indefinite from the myeloma cells, allowing for the production of the large quantity of monoclonal antibodies. Which statement about the properties of the antigen binding sites in antibody molecules is correct? The correct statement about the properties of the antigen binding sites in antibody molecule is D. They have variable amino acid sequence for different antigen. Because the antigen binding sites of antibodies are composed of variable regions that can have different amino acid sequences, and this variability allows antibodies to bind to a wide range of antigen. The statement describes way in which different types of monoclonal antibodies can work, and which type of monoclonal antibodies could be used to treat cancer. One is a monoclonal binding to protein on the cell surface and triggering the immune system, which is number 1. This can help target cancer for destruction by the immune system. And number 2, blocking molecule on cell surface that inhibit T cells. And this can help enhance the immune response against cancer cells. And number 3, blocking cell signaling receptors that trigger cell divisions. And this can be explained through, this can inhibit the growth of cell cancer. Monoclonal antibody that target and block receptor involved in immune responses are less commonly used for cancer treatment directly, though they might be relevant in some immunotherapeutic contexts. And the most comprehensive answer will be, which cover common cancer treatment, is 1, 2, and 3. That's all for today's sessions, and hope you are doing well in your studies and upcoming examination. And please don't forget to hit the like button and on your notifications to stay updated, and subscribe to Journal Study Lab YouTube channel. Thank you very much.